So at this point, we've discussed the structure of DNA as well as chromosomal structures, and we also covered the topic of DNA replication. Now we're going to discuss the cell cycle and mitosis. But before we jump in, I'd like you to ask yourself, why is the cell cycle and mitosis even in the same chapter as DNA replication? The process of mitosis we also call cell division, and that is a part of the cell cycle. When we talk about cell division, what we want to be doing is passing on an exact copy of the parental cell to two daughter cells. The first step in that process is that all the DNA in the cell needs to be replicated or duplicated, which is the process of DNA replication, which we just discussed. So one of the first things that has to happen is all the DNA within a chromosome has to be duplicated, and you get this structure seen here on the right, which is what we typically think of when we think of a chromosome. A big overview of this whole process of cell division begins with our initial parental cell, here depicted as a cell containing two chromosomes. So during the process of DNA replication, we will duplicate all the DNA in the cell. And then during the process of mitosis, we are able to separate that duplicate copy from each other until we generate two daughter cells that are identical to the initial parental cell that we started with. And when does all this happen? Well, it happens during the cell cycle. The cell cycle is known as G1S, G2, and M. G1S and G2 are called interphase of the cell cycle, whereas the M phase is the division phase of the cell cycle. G1 stands for growth, S stands for synthesis or DNA replication, G2 is another growth phase, and then again M stands for mitosis. So the whole process of DNA replication that we just discussed prior to this topic all occurs during that S phase. And this is all in preparation for generating two daughter cells from one initial parental cell. So what we're going to talk about now is this process by which a cell can become two cells. One thing to keep in mind is that this process of mitosis pertains to our somatic cells. These are all the cells in an organism that are not the reproductive cells. The reproductive cells are going to be the egg and the sperm. So when we talk about mitosis, it pertains to the somatic cells. At the end of this chapter, we'll discuss the process of meiosis, which pertains to the gametes, or the reproductive cells, also known as egg and sperm. So we need to tightly regulate this process of cell division. We don't really want all our cells to be dividing continuously. There's some cells, such as human nerve cells, that once they differentiate, they actually never divide again. There's other cells, such as human liver cells, that only divide when repair becomes necessary. However, there's a whole host of cells that should divide on a regular basis, things like skin cells that, as I mentioned earlier, we're constantly replenishing our skin cells. When we're growing, we need our cells to divide. So there's times when we want active cell division, and there's other times when you want that cell division to be stopped. And this is all regulated in the cell cycle. So again, here's a different version of the cell cycle showing the different phases. So again, as I mentioned, interphase consists of G1, which is the first growth phase, S, which is synthesis, G2, which is the second growth phase, and mitosis. And as I mentioned in the prior slide, which phase of the cell cycle consists of DNA replication, that's during this. So during the cell cycle, our chromosomes are going to look different. When they initially go into the cell cycle, we'll have one chromosome. And during the S phase, that DNA gets replicated such that we still have a single chromosome, but we now have two sister chromatids. The entire chromosome has been replicated. And students often get confused about how many chromosomes are actually in the cell. The big clue here is to just count your centromeres. The centromere is the middle of that chromosome, and that will always give you a good indication of the number of chromosomes in the cell, as long as you can just count your centromeres. Now I'd like for you to look at this cell that is shown before the S phase, before DNA replication, and what it looks like after DNA replication, after the S phase. So I have a series of questions for you that I'd like you to answer. So the first question is, are the number of chromosomes the same in cell A as what we see in cell B? The answer is yes. We do have the same number of chromosomes. Again, if you look at the number of centromeres and just count the centromeres, we have 4 centromeres on the left cell, which indicates 4 chromosomes, and on the right hand side we have 4 centromeres there as well, so we have 4 chromosomes. How about the amount of DNA? Is that the same between these two cells? The answer is no. Well, when we underwent DNA replication, we doubled the amount of DNA in cell B as opposed to cell A. How about the number of chromatids? Are those the same? The answer is no. In cell A, we have basically 4 chromatids, whereas in cell B, we have 8 chromatids, 2 sister chromatids per chromosome. So when we're talking about cell division, for any cell type, it falls within 4 key steps. The first step, shown here for a bacteria, is that the cell itself has to grow. We then need to copy the entire genome, and that's the process of DNA replication, which we've already discussed. You then need to separate those 2 different sets of chromosomes from one another, and then we actually need to physically split the 2 cells into 2 identical daughter cells. So whether we're talking about bacteria or eukaryotic cells, this overall concept is the same. It's important to note here that there is one other option with regards to the cell cycle, and that is G0, in which you actually don't even enter the cell cycle at all. G0 is if you are not in that growth phase and you do not need to replicate your cells. This would be the case for cells that are terminally differentiated and not ever going to divide again, things like the nerve cells that I mentioned in this slide before. So again, G1, you can think of it as your first growth phase. It's sometimes called first gap. S is synthesis of DNA. G2 is the second gap or growth phase. M is your mitosis and cytokinesis. Cytokinesis being that process of dividing the cytoplasm. And then again, I've listed as another option G0, which is when a cell actually doesn't enter into the growth and division at all, but instead has postponed that cell division, or if it's never dividing again. This is a quiescent state in which it doesn't enter the cell cycle. For those that do enter the cell cycle, it's important to note what's actually happening during the growth phases. So in growth 1, the cells can grow, but only to a certain point. And what we see is the cell itself, the entire cell, gets bigger. During S phase, the chromosomes are doubled, as we mentioned before, and during DNA replication. And in growth 2, that's getting prepared for mitosis. So there's some critical checkpoints that are in place to make sure that everything is set up for proper cell division to occur. The most important checkpoint is at this G1 to S transition. Here the cell is assessed to make sure it's in a healthy state, that there's growth factors around, that it's actually ready to enter into DNA synthesis. The other key checkpoint is making sure that DNA synthesis has actually occurred correctly and that the necessary growth factors are around as well before the cell enters into mitosis. So in relating the cell cycle to what's actually going on within the cell, starting at G1 in the cell on the left-hand side, what you can see is we have our cell here. It's showing 6 chromosomes. The 3 red ones are, you can assume, are from mom, and the 3 blue ones are from dad. So these are your homologous chromosomes. During the S phase, each of those chromosomes gets replicated. So each of the DNA is replicated, and you can see we still have 6 chromosomes, but all the DNA within a single chromosome gets replicated. During G2, the cell continues to grow, and then once it enters into mitosis, the cell gets ready to divide. We separate sister chromatids in the process of mitosis, ultimately generating 2 cells that look identical to the initial parental cell. So that is the cell cycle. That's the phases of the cell cycle, G1S, G2, and M. So now we can talk about the phases of mitosis. When a cell is undergoing mitosis, what we are generating are 2 identical daughter cells to the initial parental cell. There's 4 phases of mitosis called prophase, metaphase, anaphase, and telophase, which also includes cytokinesis. I always just remember this as PMAT, and that sticks with me, but do whatever you need to do to memorize it. But again, it's prophase, metaphase, anaphase, and telophase. Your book goes into excellent detail on this process, so I'm actually not going to discuss it any further in this video lecture. Refer there for all the key steps and what's happening in each of those phases. You just need to have a good working knowledge of what's occurring. You don't need to memorize every aspect of every phase. So now I have a few questions for you just to see if you grasp the information. So which term best describes the structure circled below? Would you call that a single chromatid, a single chromosome, or a homologous pair of chromosomes? I'll give you a second to answer that. The correct answer is a single chromosome. As you can see, this structure here contains 1 centromere, so that's one clue that it's a single chromosome. What gets separated at the metaphase plate in the next phase, you would see, is a separation of those sister chromatids. But as of right now, there's 2 chromatids there. And we're not separating homologous chromosomes. That's something that happens in meiosis. Next question, the 2 circled structures are exact copies of each other. Is this true or false? I'll give you a second to answer that. The answer is A, true. The 2 structures that you see circled are identical copies of each other. That's the whole point of mitosis, is by the end of this process, we'll have 2 cells that are identical to one another. So this is the step in which that replicated DNA is getting separated from each other. So in summary, for the cell cycle and mitosis, eukaryotic cells need to divide for purposes such as reproduction, development, repair, and replacement. Prior to the actual physical act of division, the cell has to duplicate its entire genome, which is DNA replication or DNA synthesis. Somatic cells are diploid, while our gametes, the sex cells, are haploid, and we'll talk about that more at the end of this chapter when we discuss meiosis. Mitosis is a very orderly mechanism for equally dividing the cell's duplicated genome and splitting the cytoplasm into 2 daughter cells. So in conclusion, the cell cycle includes the G1S, G2, and M phases, and that M phase of mitosis can further be broken down into PMAT, prophase, metaphase, anaphase, and telophase. I hope this helped. Take care.